Hello, everyone. I'm Jeffrey Snook Cosgrave, and today I want to talk about unlocking the magic of Lactobacillus, especially when it comes to brewing exceptional sour beer. Lactobacillus is a genus of bacteria that has existed for thousands of years. Comprised of various bacterial species, it serves as a broad term encompassing lactic acid producing bacteria. These bacteria generate lactic acid by metabolizing carbohydrates and sugars into lactate, a topic we'll explore in greater detail later. Lactobacillus bacteria are gram-positive, typically rod-shaped, and fall under the Firmicutes phylum. They are generally recognized as spoilage microorganisms due to their role in acidification and the production of unpleasant odors, particularly noticeable in the context of beer production. Lactobacillus, a well-known beer-spoiling microorganism, is currently revolutionizing the craft beer industry, and not in the way most would expect. Although Lactobacillus is typically not a desirable addition to beer, some brewers have discovered innovative ways to turn challenges into opportunities, such as lambics, Berlin Weiss, wit beer, and American sour varieties derive their unique flavor profiles from the deliberate inclusion of Lactobacillus during the fermentation process. The lactic acid produced by Lactobacillus imparts a distinct sourness to these brews. Sour beers have been gaining popularity in the United States, particularly fruited sours, which involve adding fruit to provide both additional sugars for higher alcohol by volume (ABV) and organic acids for a more pronounced tartness. In 2021, Untappd, a popular beer ranking app, ranked fruited sour beers as the nation's sixth most popular craft beer. Lactobacillus is often grouped together with Pediococcus bacteria because both types of bacteria can produce lactic acids, contributing to the sour flavor profile of beer. However, a significant distinction between these two microorganisms in the brewing process is the time they require to achieve the desired sourness. Lactobacillus has the advantage of speed, capable of accomplishing in a week what Pediococcus would take a year to achieve. This rapid lactic acid production is why Lactobacillus is often associated with spoilage. However, this speed comes with a trade-off. Beers soured with Lactobacillus tend to lack the unique and intricate flavor profiles that can be achieved with Pediococcus. Nevertheless, in today's popular fruited sour beer styles, the most favored type of sour beer in the US, this distinction becomes less significant. This is because organic acids such as citric acid from the added fruits are used to enhance and intensify the tart flavor profile. Understanding Lactobacillus. Lactobacillus accomplishes the production of lactate through the fermentation of glucose, a process orchestrated by the glycolysis pathway. Lactic acid can be generated from both 5-carbon sugars, pentose, and 6-carbon sugars, hexose. This versatility in utilizing glucose for lactic acid synthesis underscores the commercial significance of Lactobacillus. In commercial applications, Lactobacillus holds great value due to its notable tolerance to high acidity, its ability to yield substantial quantities of lactic acid, and its overall productivity. The production of lactic acid and the bacterium's productivity are strongly influenced by environmental conditions. Lactobacillus thrives within specific pH and temperature ranges, varying among different strains, of course. Furthermore, Lactobacillus exhibits diversity not only in its environmental requirements, but also in its metabolic outputs. Some Lactobacillus strains exclusively produce lactic acid as their primary end product, while others generate a variety of end products alongside lactic acid. This distinction categorizes lactic acid producing bacteria into two classes, homofermentative and heterofermentative, while even some Lactobacillus species are able to encompass both classifications, contributing to unique characteristics that brewers can harness when crafting sour beers. The Big Three. There are several different strains of Lactobacillus commonly employed in the brewing process. However, let's delve into three of the most popular Lactobacillus strains used in sour beer production, each possessing different characteristics. Firstly, we have the heterofermentative bacterium, Lactobacillus brevis. This strain is prized for its ability not only to produce lactic acid, but also to yield other byproducts like ethanol, carbon dioxide, and acetic acid. It's the presence of acetic acid that imparts a vinegar-like flavor profile to the beer, a sought-after trait in many unique sour beers. Next up is the homofermentative bacterium, Lactobacillus delbrueckii. This lactobacillus specializes in lactic acid production as its primary end product, offering simplicity and precision to the brewing process. Lastly, we'll explore Lactobacillus plantarum, known for its facultative nature. This means it can adapt its behavior, shifting between homofermentative and heterofermentative modes based on prevailing environmental conditions. As you can see in the graph down here, I'll see if I can pull this up, they all have different pH that they enjoy. You can see that Brevis and Plantarum seem to be around the same, while De Brucki is way up here. And it's even noticeable on this graph as well. And I thought this picture was funny because you have Brevis, Plantarum, and De Brucki in the same photo. How unique, all in the incubator. Okay, before we can continue, we need to take a time out and look at hop sensitivity. So let's delve into the significance of hops and their role in the brewing process with a particular emphasis on their remarkable capacity to deter Lactobacillus and other spoilage microorganisms. Hops represent an integral facet of the brewing process and were enshrined as one of the three fundamental ingredients in the Bavarian beer purity law of 1516, the Reinheitsgebot, alongside barley and water. It's also worth noting that yeast, which plays a vital role in fermentation, was not yet understood and was believed to just magically show up, but really it was insects feeding off the mash that introduced the yeast to the whole role. So hops, primarily through the compounds they contribute to the mash, serve as a potent inhibitor of Lactobacillus growth. This inhibitory action is chiefly attributed to the isoalpha acids produced by hops. These isoalpha acids possess antibacterial properties that are particularly effective against gram-positive bacteria like Lactobacillus. They function as ionophores. If we remember those, they're essentially ion bearers by disrupting the pH gradient across the membrane of Lactobacillus cells and diminishing the proton motive force. Consequently, this disruption adversely affects the proton motive force dependent nutrient uptake, ultimately leading to cell death of our gram-positive bacteria. It's important to recognize that throughout history, hops were routinely incorporated into beer production to extend its shelf life, preventing spoilage, and to ensure longevity. Continuing on with our timeout, I wanted to show a great example of this practice being the Indian Pale Ale, or IPA. The IPA, a traditional pale ale, distinguishes itself by featuring a significantly higher hop content, resulting in increased alcohol content and bitterness, as well as enhanced resistance to Lactobacillus contamination. This strategic use of hops was vital for British brewers, as it allowed them to craft a robust and bitter ale capable of surviving the lengthy voyage from British to India, circumnavigating the Horn of Africa, where the extended journey often led to beer spoilage, primarily due to Lactobacillus contamination, most likely Lactobacillus brevis, by the way. To counteract this challenge, the British augmented the hop content of their pale ales, grounded in the belief that hops possess preservative properties. In the end, this practice gave rise to the iconic IPA, characterized by its bold bitterness and resilience against spoilage during the arduous sea voyage. And as you can see here, they had to go all the way from Britain around the Horn just to get to Africa, and the pale ales with less hops were just not making it. They added more hops to it, and they were able to survive all the way to India on that boat. All right, time in. Let's get back to Lactobacillus. The first we're going to start out with is Lactobacillus brevis. It is classified as heterofermentive, meaning along with lactic acid, it generates additional byproducts that can influence the character of the fermented beer. While Lactobacillus brevis lags behind in its homofermented counterpart, Lactobacillus delbrueckii, in lactic acid production, it distinguishes itself by independently producing ethanol. Furthermore, Lactobacillus brevis exhibits a degree of resistance to hops, making it conducive to the crafting of sour beers with higher alcohol content. This capacity for autonomous ethanol production, coupled with yeast interaction, contributes to the elevated alcohol content observed in sour beers fermented with Lactobacillus brevis. Remarkably, Lactobacillus brevis also demonstrates a moderate level of tolerance to hops, allowing it to thrive in a mash that contains these bittering agents. It's this hops resistance that has somewhat tarnished Lactobacillus' reputation as a potential spoilage organism, as it can proliferate in conditions where other lactobacilli might struggle. In terms of fermentation temperature preferences, as we saw in the graph before, Lactobacillus brevis thrives within the ranges of about 102 to 105 degrees Fahrenheit, making it well suited for specific brewing conditions. Continuing on, let's look at Lactobacillus delbrueckii. Lactobacillus delbrueckii boasts a remarkable ability to produce lactic acid as its primary metabolic product, surpassing other heterofermentative bacteria in this regard. What sets Lactobacillus delbrueckii apart is its single-minded focus on lactic acid production. This specialized approach allows Lactobacillus delbrueckii to yield approximately twice as many lactic acid molecules from a given quantity of carbohydrates compared to other heterofermentative lactobacilli. This unique characteristic, coupled with its homofermentative nature, has rendered Lactobacillus delbrueckii a popular choice not only in brewing, but also in the production of dairy products. However, it's worth noticing that Lactobacillus delbrueckii operates optimally at a fermentation temperature of approximately 110 degrees Fahrenheit, with a range of about 110 to 115, and if you can remember from our graphs previously, that is a little bit higher than both brevis and plantarum. In the context of sour beer production, it's important to highlight that while Lactobacillus delbrueckii excels in lactic acid production, it does not possess the capability to generate ethanol. Consequently, the presence of yeast is essential in the fermentation process to provide the desired alcohol content. This interplay between Lactobacillus delbrueckii and yeast can affect the alcohol by volume of sour beers brewed with this microbe. Typically, these beers tend to have a lower ABV compared to those fermented with other microorganisms. ABV is alcohol by volume, by the way, if I didn't mention that, as Lactobacillus delbrueckii and yeast vie for the available glucose in the mash. Moreover, it's worth noting that Lactobacillus delbrueckii exhibits limited tolerance to hops and typically thrives in an environment with an international bitterness unit, IBU, of less than 5. And such a low IBU renders the inclusion of hops as an ill-advised idea, as it would impact the flavor profile below the detectable range for most human taste buds and just be a waste of money. Finally, let's look at the last member of the Lactobacillus family I want to talk about today, Lactobacillus plantarum. What sets Lactobacillus plantarum apart is its remarkable ability to seamlessly transition between homofermentative and heterofermentative modes depending on the prevailing conditions it is in. This inherent flexibility is what makes Lactobacillus plantarum an indispensable player in the brewing process. It can adeptly navigate under wide temperature variations while effectively reducing the pH during fermentation. It's worth noting that Lactobacillus plantarum's exceptional characteristics have led to its recent reclassification as lactic Bacillus plantarum. Nonetheless, its distinctive facultative nature and its prominent role in the brewing industry make it a valuable addition to our discussion. It is also widely used as a use-all Lactobacillus in the brewing process. In summary, Lactobacillus bacteria, with their ability to produce lactic acid, are transforming the craft beer industry, particularly in the realm of sour beers. While traditionally considered spoilage microorganisms, some brewers have harnessed their unique qualities to create popular beer styles. Variations within Lactobacillus strains, such as Lactobacillus brevis, delbrueckii, and plantarum, offer distinct advantages and characteristics, adding complexity to the brewing process. Additionally, hops play a crucial role in preserving beer by inhibiting lactobacillus growth, as exemplified by the historic development of the Indian Pale Ale. This dynamic interaction between microorganisms and ingredients underscores the ever-evolving world of craft beer production, shaping a diverse range of flavors and styles. Thank you again. I'm Jeffrey Snook Cosgrave. I actually forgot that I have one more slide, but here are my sources in case anyone wants to check out any of the research I did. And thanks for watching the video. I really appreciate it.